Hello guys, my name is Dr. Varun and you are watching Chlorophyll Classes only on YouTube. Well, this is for 12th class. This episode, we will be talking about all the other things related to genetics. Well, not completing the chapter in this video as of now, but definitely we will complete it before the end of June. So let's just start with this. Till now, what we have studied? We have studied about the Mendel, post-Mendel genetics, something different than what the Mendel has told. Let's just see that why the Mendel work was not appreciated by the time. Well, Mendel published his work in 1865. Okay, so Mendel published his work in 1865. Okay, when he was working on it, it was close to that. Now what happened during that time, lot of problems were there. First of all, there was not much of communications that was built. Okay, so when the work can't be flowed to the major people, to the major society, they are not getting enough recognitions, right? So for example, I am your teacher, Chlorophyll Classes is there. Until we will be on 192 subscribers, we might not be able to reach to the much people. Same thing happens with the Mendel. So right now, me, Varun sir, is your Mendel only. Okay, so let's just go back. So what happened? So first problem was due to poor communication. Due to poor communication, Mendel work was not spread wide. Okay, first problem. Second problem was what Mendel used to talk about genes, which is in his word is about factors. Right now we know they are genes, but he calls them what factor as a discrete unit of inheritance. But when we see from the other point of view, like from other plants, organisms, it is not like he said that it goes very what we say that without any contamination, it will be dominant, it will be recessive, but we never saw those things in real things were little more different, and he's not able to tell the things because he don't have the knowledge of a lot of things. So second problem was Mendel consider gene, which according to his word was factors, as discrete unit, means these are those particles or packets which are actually being inherited and giving the properties. Okay, and they're in the pure form. Okay, so you know the law of purity of gametes, second law of segregation of gametes, right? Discrete unit as unit of inheritance, as unit of inheritance. Okay, and the third one says that after this what happened, that Mendel uses mathematics in biology, like the phenotypic ratio. This will be the ratio of the plants that will come in this property and all, which was also not acceptable at that time. The next thing was use of mathematics in biology was, you know na, that they used mathematics in phenotypic ratios. They are able to find it out how many will be this, how many will be this, how many will this come, right? Use of mathematics in biology was unacceptable at that time. And the fourth one is, it was the era of Darwin. Everyone was busy in talking about origin of species, Darwin theory of natural selection and all those things. Okay, next thing, it was era of Darwin. Scientific community was busy in discussion of origin of species and those concepts. So that led to what Darwin work put into sight. Okay, and this leads to and this leads to sidelining of Mendel's work. But now other things were taking place. Soon microscope was made, and by 1900 three independent people rediscovered the Mendel work. And you know that 1865 to 300, almost more than 35 years has been completed, and people would be like, oh sorry, 45 years would be completed, and there was so much of advancement in the technology. Microscope is also there, almost at the point of like we have made it the basic microscopes, so we are able to see more things. Okay, and when we see more things, we found out that something called as chromosomes, which are inheriting in the similar pattern the Mendel said. And this Mendel work was rediscovered by three different people. Okay, so means Mendel is died by now, but his work in 1900s, three scientists rediscovered, means they discover something and they were like, oh, this is something very interesting, but then they will be like, okay, let's see that back then when they trace it, they found that Mendel has already done this work. They were like, shame man, our Nobel Prize has been lost, something like that they said. Of course, I'm just giving you an example. It's not happened like that. So they were like, oh, they have all, like Mendel has already done our work, which we have worked so much of our time and investment. But it was important that they rediscovered it, because this leads to what? This leads to the thing that Mendel work was recognized again. Genetics has something which is true. What has Mendel has said that it has some way in it, right? So three scientists rediscovered the Mendel work. Now you have to remember the name of three scientists. Number one is Hugo de Veris. There is Corinus, and there is von Tishmark. These three rediscovered it. As they rediscovered it, with the fine, like of course the microscopes were also being made, and we are able to understand the cell division is taking place, and how during the meiosis there is a doubling of factors are there, how they are, like we used to double, they will go 4, they will go, and that's why this structure is coming up, like this Punnett square has been made. That has a proper evidences. So what has happened? So if you know about the cell division, okay, then you can understand the facts. Okay, so what was the fact? Well, here I will try to make you explain if you don't remember in the 11th class, because I know that those students who are in 12th, who has not done their 11th very well, they are struggling with this. But don't worry when the Varun sir is here, right? So let's just see that. So what happened? And in 1902, through microscope, cell division was also studied. With advancement in microscopic technology, complete meiosis is also studied, and it leads to two things. Number one is what? That darkly stained body in double form, sorry, darkly stained body called chromosomes are found, which works like factors. Of course, we will understand much better. Just remember the fact. Moreover, it also stated that these chromosomes during meiosis shows doubling up and hence verify the Mendel F2 generation results. For now, just remember the fact. Okay. So let's just see that what is chromosome and what is gene. Gene means what Mendel talked about, factors. Okay, we know right now they are genes. Okay, let's just try to dig deep into it. So if I see the chromosomes, always remember in the chromosomes will appear when the cell division will occur. Okay, how they will occur, we will learn in the molecular basis. We will understand it in the molecular basis. Here you remember that chromosomes would be what? They will be in pair. Okay, but what homologous pair means, till now whenever you have seen certain diagrams that this is a chromosome number 1, 2 chromosomes are shown to you, means that a pair is being shown to you. Normally in 10th class also you might have, when you were studying about genetics, you might be doing what? The sex determination in humans, right? XY type. So we said that we have what? 23 pairs of chromosomes, right? So what we learned from till now is that the chromosomes look like this. Okay, what is this? We will learn more better in the molecular basis chapter. The next chapter we are going to be starting from July. Okay, so this is what chromosomes. Always remember beta, chromosomes will be in pair. Okay, so chromosomes are what? They are paired and both would be chromosome number 1 only. Example, I am taking that example. This is chromosome number 1. So why I have 2 chromosome number 1 in each of my cell? Because beta, one is coming from my mom, one is coming from my dad. That is how genetics started. Na? There is a male parent, female parent, and then it is coming in the baby. Okay, those genes like RR, remember, this way. So here what happens? So these are the chromosomes. These paired chromosomes are homologous chromosome. Homologous chromosome means similar chromosome with a similar structures or things present on it. Now on this chromosomes, chromosome is what? Genetic material. On genetic material, what is normally present? The things which it has to show, right? All the genetic material will be what? For example, this whole DNA is there. Every part is telling one thing, that this part is telling that how good my mind would be. This part telling that how I will speak, what would be the tone of my speech. This part telling this part telling how my eyes, this part my high neck. Different chromosomes doing different things, right? Now in this chromosome, those different parts which are doing different characters, those characters are provided by genes. So on this chromosome, what is present? There is the genes present, the factors present. So when I'm making it with the yellow means this is one pair of alleles. Okay, this is different thing. So I will write it. This is what? One gene. And since this gene has what? One part is present on this, one is this. They are what? They are the alleles. Alleles means it's type, contrasting characters. Remember dominant and recessive ones? These are what? Alleles. If you remember that, what we have studied in the first chapter terminology, sorry, first part terminologies. So what are they? They are the different allele of it. So this is first allele present on one chromosome, and this is the second allele of one gene, one number gene. Okay, so this thing you have to remember. So now you understood what is allele. Alleles are the contrasting character of the genes, and genes are what? These are the certain locations present on it. If someone will ask you that how these genes are made up of, these are what? These are group of nucleotides. Okay, what are nucleotides? We will learn in the molecular basis. Here just remember that we have different nucleotides present. They come together and start acting like one unit. Okay, so how they will do it, we will learn it later. Just remember over here that certain nucleotides are forming what? Genetic material, and those genetic material will make what? It make a gene. Okay, gene has two different forms. In this, same could be present, different could be present. On one chromosome, there are multiple genes present. Like this is another red color one is different. Then comes this green color. Green color one is different and taking place of different thing. This blue color one is different and taking care of the different character. So multiple genes are present on the chromosomes. But one thing we can understand is that if we have to draw the similarities between chromosome and genes, what could be the parallelism? Okay, and what could be the difference? That is the very important. So one thing we know that genes are also in the pair. Genes are present on the chromosomes. Chromosomes are also pair, but chromosomes are present inside the nucleus. Okay, so this thing we're gonna be writing it up. What is the parallelism between okay, parallelism between genes and chromosomes. Okay, so first parallelism is what? Both occur in pairs. Okay, both lie in paired condition in all diploid cells. Remember, we are talking about the diploid cell in the chapter most of the places. Okay, so accordingly we are working. Okay, throughout the initial I have told you what is diploid and haploid. If not, I will be soon uploading a short on it, you can refer to it. Okay, then the second thing is that both are expressed in the paired form only. Single, if they are present, not able to express. Okay, so this you have to remember. So means if one chromosome only from my mother side is coming, father side is not coming, not able to express. That's why the gametes are not the human body. They had to be in the diploid form. Okay, so with respect to diploid organisms, okay, both are able to express in paired condition only. Next is during the formation of gametes. This is very important because this thing, when we understood in the chromosomes, then only we said that the law of segregation and law of independent assortment is correct, which we will be coming up after this only. Okay, so it says that during the during the gamete formation, what gamete formation? Your chromosomes separate. Okay, if you don't know, you have to remember that since only half of the chromosome has to be go into the gametes. Okay, because if everyone will go, then what will happen in the next progeny? Extra chromosomes will go. Half will come from mother, half will come from father. So means when the gamete has to be formed, half of the chromosome is needed in my body and same in the female body. Okay, so what happened? Chromosomes separate from each other, homologous chromosome. Similarly, genes also get separated. They can't go together because chromosomes, they are present on the chromosomes and they will be eventually separated. Okay, so during gamete formation, both are reduced to half. That is, single chromosome with a single gene will go into the gamete. Okay, that is single chromosome. So did I say single gene, single allele? Okay, single chromosome with single allele of a gene enters in a gamete. And what will happen? Gamete is formed. When male gamete and female gamete will fertilized, means fuse, what will happen? Again, they come into their paired conditions. Okay, so this is what independent assorted themselves and they come into the paired form. Okay, so what happened during fertilization? During fertilization, paired condition of both genes and chromosome is restored. This fact you have to remember, and rest I have told you about where will be present, where it is present. Okay, so these genes are present on chromosome. One chromosome, multiple of them present. Okay, this thing, if you will remember it, very good. Let's just move forward towards the chromosomal theory. Okay, now they see that okay, this is happening and everything is there. They gave a chromosomal theory. Okay, so in a chromosomal theory, what happened, that it is, first of all, remember that given by what? It was given by Sutton and Boveri. Okay, so Sutton and Boveri told about this chromosome things only. Okay, let's just study one by one about them. We will understand those things and then see that how they are explaining the facts. Okay, how the chromosomal theory is with regard with the Mendel theories. Okay, similar to it. So our heading is Chromosomal Theory of Inheritance. So first thing is what? Given by, proposed by Walter Sutton and Theodore Boveri. Okay, they can just give you this Sutton and Boveri, that is also correct. Okay, now what they are saying, let's just see. Okay, one by one. Number one thing, the facts they are saying that chromosomes, okay, how they are lying and how the things are present, whatever we have studied till now is being told to you in detail. So if I have to see it, I will go with like, it says that genes and chromosomes, they lie in pair. We know that, we have seen in the diagram also, understand it. In the somatic cells or in the vegetative cells, means that those cells which are not reproducing. In a reproductive cells, they in either one of the pair will be present into it. And allelic pair, or in the chromosomes, they are like genes lies in the allelic pairs, means two different alleles would be present on chromosomes. Which chromosomes? Homologous chromosomes, means which are similar. It won't be that one chromosome is chromosome number and then other is chromosome number 2 in the female. That never happens. The basic nature of the genetic material in all the human beings remains same. What a difference would be there? That there will be allelic differences that can occur, nothing more than that. First point is clear. Can I write this first point? So the first point says that genes and chromosome lies in pair inside somatic or vegetative cells. Okay, genes lie in allelic pair on homologous chromosomes. So chromosomes will be in what pair? Homologous pair. Okay, homologous chromosome. Now the next fact is, in each homologous pair, one chromosome comes from male, other comes from female. Okay, so in each homologous chromosome, one chromosome comes from male parent and which is known as paternal chromosome. Okay, known as paternal chromosome, and another chromosome comes from female parent known as maternal chromosome, known as maternal chromosome. Okay, now during meiosis, gamete formation in the somatic cells, means in the the mother cell, okay, whatever the sex mother cell we can say, okay, they will segregate. Okay, so pairs also segregate, allelic pair also segregate, and the homologous pair also segregate. Okay, so same thing is repeated again by them, but we're gonna be writing it up. So during meiosis, meiosis will occur what? When the gametes will form. Gamete formation, a single chromosome and allele enters a gamete from the pair. Okay, means not the pairing is not going. Okay, from the pair. Next thing is that neither chromosome or allele is able to express itself in alone form. So means gamete will not be able to express and grow. Okay, so neither chromosome or allele is able to function in single. Next is normal chromosome number or the ploidy level in a somatic cell is restored when the zygote is formed. Okay, so normal chromosome number or ploidy level is restored in zygote after fertilization. Okay, I hope these are the facts which we have already discussed. Now, on the basis of chromosome, like the question that will come in your board exam is that justify or explain the Mendel law in accordance with the chromosomal theory, or how the chromosomal theory is telling about the law of segregation or law of independent assortment is true, or verifying it. Let's just see that. So what happens in that? So this is what first thing we are learning. What? Explain how chromosomal theory, explain with the help of chromosomal theory. Explain, because till now we said that okay, gene and chromosome has parallelism, so Mendel has talked about something, and let's see that how this chromosomal theory is in accordance and explaining that thing also. Okay, so we are, because what, the work has been refined, like rediscovered, right? So this rediscovered work, if has a similar thing which was already present, it should justify, right, those things with this thing. Means these are what? Experimental proofs. At that time he don't have the experimental proof, Mendel. This is one of the other problem also, so we can write it also. We will write it. Okay, one more point is that no experimental proof of factors. Okay, so here we are coming. So explain with, okay, this should be like explain law of segregation with help of chromosomal theory of inheritance. So let's see that what is happening. If you see over here, they are what? They have made different chromosomes. So if you remember the cell cycle, what happened when the cell is there during the S phase, it doubles. Then G2 phase also, it will keep on double. Then the meiosis will occur. In meiosis, what will happen? Prophase, metaphase, anaphase, telophase will come. Now in prophase, what happened? There is, if they needed a crossing over, if the meiosis is taking place, crossing over will occur. Then what will happen? Then there will be metaphasic plate, then they will divide, and then they will form this, and then further again meiosis 2 will occur. Right, let's see that here the gametes are being formed, how it is being done when the gametes are being formed. So you should remember about the meiosis. So let's say that this is G1 phase. During G1 phase, what will happen? These chromosomes are present. Now what will happen? The chromosome increase their number. G2 phase, because during S phase, what happened? Doubling of the genetic material occur. Chromosome remains same, because this is what? One chromosome, one chromosome. Here also one chromosome, one chromosome. But here what happens? This chromatid is extra. Okay, I hope that you know that what is chromosome, chromatid and all those things. If not, I am telling you briefly over here. What happened, beta? This chromosome is like this. But during the time of meiosis, what happens? Cell division, they increases their number. Now earlier what has happened? This is what? This is called as one chromatid. This is, it's like a, in one chromosome. Okay, this is what? One chromatid, another chromatid. So two chromatids are there in this case. Okay, so when I'm saying chromatid means what? It is related to one chromosome. There are two chromosomes, they both are homologous chromosomes. Okay, I hope this thing is understood. If not, then let me know in the comment section. I will help you out with this. Okay, or with the 11th class, when the cell division chapter would be there, these things will be discussed over there. So what happened? They increases their number. Okay, now what will happen when the meiosis 1 will occur? This meiosis 1, what will do? It will cut these things. So what will happen? This thing will be cut, this thing will be cut. So what happened? This cut and this cut, they come together. So they becomes one cell, this become another cell. Of course, these two are what? These are the homologous chromosome, and these two are the homologous chromosomes. Why the color is different shown to you? The color is different shown to you so that you can tell that different genes are present on it. Okay, so that's why they are showing it like this. So if I see that this is what? This is one pair of chromosome, and these are what? These are second pair of chromosomes. Now bivalents are what? Bivalents are that their chromatids have increased to double. So that is bivalent case. So here those bivalent cases there. Now what happened? This first chromosome, like this is the first chromosome. It's one thing goes with this thing goes with this. So this is how it went. And further they will divide and gamete cell will form like this. Okay, so this thing has been shown to you. Now what they are showing? They are showing that irrespective of this has this part, means it's homologous counterpart, but they are not influencing. They are easily going into the different, just like the gametes. So if I have to write it and explain you little more, see over here what is happening. If this is A and this is small a, this is B, this is small b. So what will happen? This A and this small a, this capital B, small b, because they have increased their number. Why they are increased their number? Because Punnett square. If you remember that we used to make 4 gametes. So it will make 4 gametes. So at the end, let's see, this 4 gamete cells are there. So how they will make? By doubling themselves initially at the S phase. Okay. So now what will happen? They are dividing. So here also A is there, B, small a, capital B, small b. So in first case, what happened? This divided like this. So it has what? Capital A, capital B, small. So the next cell that form is what? AB, small. So here what happened? 4 cells form from initially this one. Okay. So initially the parent was what? If this is the parent has this genotype. Now this genotype, we have learned it. If you have gone through my all video, I have told you that how many gametes will it form. With the Forklian method, we can do it. What we used to do, we used to write like this: capital A, small, capital B, small. So this is capital, small. So what happened? One gamete will be AB. Another gamete would be what? Capital A, this B. This can be this can be this. But these are taking place when there is what? New thing are being formed. So 4 gametes will form. 4 gametes are formed here also. Of course, these gametes has option. What option? That this part can go with this, but this part can go with this either. New recombination. If this part has gone with this, it has fully opportunity to do it. Okay, so this will do. So that part will be the independent assortment, I will tell you. Here I am just telling you that they will segregate like this. 4 gametes will form. Whether be same or different, that depends upon. Okay, let's just see this thing. So this was the thing. So let's just explain those things and I help you the write-up portion with it. Okay, so the first thing is what? The first thing is at the time of the gamete formation. So I'm writing it up and explaining it to you. So at the time of gamete formation, homologous chromosomes bearing the factors are what? Genes are segregated. Segregated means separated. See, these homologous chromosomes are separated. So this is one chromosome, this was another one, they are separated up. Okay, so this thing, okay, are separated in anaphase 1. So if you see that during this anaphase 1, they will separate into this 2 formation. Okay, anaphase 1 of meiosis 1, producing dyad of the cells. Dyad means 2 daughter cells. Okay, so I am going to be writing it 2 dyads of 2 daughter cells. Okay, now they will further go and forms 2 more. Okay, so next thing is in anaphase 2. In anaphase 2 of meiosis 2, the 2 chromatids, because within one chromosome, what is happening, beta? See over here that there are 2 chromatid present. So they will also separate. This thing will go this thing will go different in this case. Okay, so what will happen? The 2 chromatids of each chromosome, means each cell's chromosome, separate and enters the 2 new daughter cells. 2 new daughter cells. Now these 4 cells, total means 4 cells has been formed. Okay, so these 4 cells, these 4 daughter cells thus formed have 1 chromosome, has 1 chromosome along with 1 allele of a gene. Of course, there will be multiple genes, but here we are talking about 1 gene so that you can remember it. 1 gene or factor present on it. Thus, the daughter cells are haploid and known as gametes. Okay. Thus, daughter cells are haploid and known as gametes. So this is one thing, one explanation we get. But what about the taking up? So I have told you over here only that it has options. What option? That this one thing can go with this or this thing can go with this. So option comes up now. I have shown you also. So this thing is explained over here, that how the law of independent assortment is also being explained over here. Let's see that. So what possibility is there if we see that? First we will see it and then we're going to be writing it up. Okay, so possibilities is like this, that this is the chromosomes. These are what? One pair of homologous chromosomes. So I'm going to write it. So this is one pair of homologous organs, homologous chromosomes, and this is second pair of homologous chromosomes. So in first case, what NCERT is showing? NCERT is saying that they goes like they separated up, they choose this, they choose this, this choose this, this choose this, just like the first one. So what happened? This options comes. So they will further divide and 4 cells would be like this. Means or like 2 different types of cells will be formed, gametes will be formed. But what happened? This goes with this, but this goes with this pair. This goes with this, but this goes with this pair. So this only they have done. So first they had done what? Like they have made it a little bit different, that this whole thing has an option to go with this. This whole thing went with this. Anything can happen. So they have showed you that this don't have to just stick with this. They can go with the other chromosome part of it. So means maybe this is the mother part, mother chromosomes, and this is the father chromosome. So first mother chromosome came, but this father chromosome came. So here the option comes up. I hope this thing is understood. See yourself and understood it. Why this green thing here and there? What is coming up next? Let's just help you out with writing it up. So options were there and we're going to be explaining it up. So what it is? Explain law of independent assortment with help of chromosomal theory of inheritance. Okay, let's just do it. I'm gonna write it up on the next page so that everything could be written up. Okay, so what it is, it goes like, in the figure we have to keep on saying. Okay, so I'm putting the figure on one side and I'm gonna be writing up the things so that you can understand this thing. Okay. Okay. So if we see, okay, let's just put the figure. Split. Okay, so this is the figure. So let's just see. The first thing, the figure 1 which is present. Okay, so possibility 1 we're going to be talking about. Okay, so figure 1 explains how it is explaining the Mendel law by the genes movement, how they are taking place. Explain Mendel's law of independent assortment in terms of movement of chromosomes during meiosis. Okay, I am not telling you that they will take place after, what choices they have, prior to only what things they will do it within when they are doing in my body itself. Okay, so when the gametes are being formed in my body. So means within organism, body, gamete formation time. Okay, so I have just write it into the bracket so you remember that reference at what point is going on. That is the most important thing. Now these are present in these chromosomes. There is present of 2 pairs of genes on 2 different pairs of chromosomes. Ideally, I am taking it up, okay, on 2 pairs of chromosomes. Then allelic genes of seed color, like for example, I am taking this, what they have taken the seed color, okay, okay, anything they can take it up, okay. So for example, I am taking it with the seed color, okay. So what happens? Allelic genes of seed color, what would be the seed color? Yellow and green, are present on one homologous pair. That homologous pair color, because when I'm talking about the chromosomes, these chromosomes are being shown. Alleles are being shown with the help of what? Green or yellow color. Okay, means this homologous pair means orange, yellow. So whatever is present is on this. So imagine that, okay, this color don't justify. So we're going to be going with this. So imagine that these chromosomes are there. So this is the factor or gene which is present. So we have considered one thing. Next thing that we are considering would be what? Then allelic shapes. So allelic genes of seed shape. So seed shape could be what? Round and wrinkled, are present on one homologous chromosome, and this time red and green one. So this red and green chromosome number is this one. So here what I am doing, so on this which I am making, this is another factor present, means the factor which I told. Now what options are there? Either they go with this case, the first option, or in this case what happened? They went with this. So means twisted happened. This is only we're gonna write. That one case it has option that it can go with this, or in this case it has option with this. That's it. Nothing fancier I'm gonna write. That's it. Okay. So what gonna happen now? Now parents homozygous for yellow and round seeds. So I am taking the corrector. Okay, seeds produce gametes which have one chromosome of each type, and thereby they have one gene of yellow color, that is capital Y, and one gene of round shape, that is capital R. Okay, simultaneously the other parent. Okay, so similarly parent homozygous for green and wrinkled will have green gene small r and wrinkled gene that is small r. Clear? Now so yeah, green will be y, small y. Okay, now when the cross pollination will occur, cross pollination will give F1 generation as 100% this. Now the F1 will produce the gamete. That makes the problem. Okay, so now when F1 produces gamete, then 4 different types arises. That is capital, small, and this. Okay, and this leads to progeny in F2 in ratio of 9:3:3:1. So means you have to explain what? The dihybrid cross. With this, the chromosomal theory expression that segregate differently and then they produce something differently can form. NCRT has just showed you this one, but I have even explained you that the option is there that they can half go with this, half part go with this. So 4 different options will come. Well, that's all for this video. Sorry, my clothes are changed because I forgot that this part I haven't recorded in the last video and I had to go somewhere else. So that's why the clothes are changed, but this is a part of the video. So if you like this video, give it a like, share it with your friends. Don't forget to subscribe if you haven't. It motivates me a lot. And the notes for this chapter would be available on the Telegram or on my WhatsApp channel. The links would be available there. Their link of my Telegram channel and WhatsApp channel will be in the description box, so you can find your queries or questions and everything over there. Well, that's all for this video. Thank you.